Hello everyone, welcome to my presentation titled Searching for Fluorinated Compounds in Central Florida Plants. My name is Cade Roberts. I go to UCF. This is for the Seminar 2 class, and I work in Dr. Elsheimer's research lab. Here we have the general outline of the presentation. We're going to start by talking about the properties of fluorine, why we're interested in it. We'll move on to fluorinated compounds that have already been found in nature, and we'll outline the general research process, gathering material, extracting, using NMR to analyze it, which species we analyzed, and then what we would do moving forward now that my research is done. So here we have some properties of fluorine. Dr. Elsheimer's lab is primarily focused on organofluorine research. There's not a lot of naturally synthesized fluorinated compounds. A lot of organisms have trouble incorporating fluorine into their metabolisms because it is the most reactive and electronegative element. There's a lot of collateral damage that occurs when organisms attempt to incorporate fluorine into their systems. This happens even though other halogens are very common in naturally synthesized molecules. We could not live without chlorine or iodine, but fluorine we have a tough time using. Even though it is the 13th most abundant element in the crust of the earth, organisms still have not found many ways to incorporate it into their systems. So now we'll have a quick summary of all of the naturally occurring fluorinated compounds that have been discovered so far. There are not very many of them. The most well-known example is a molecule called monofluoroacetate. It is found in certain species of plants throughout the world, mainly from one family. It is similar in structure to acetyl-CoA, which we know is an integral part of the citric acid cycle. The structures are down below. It's produced by the plants as a deterrent to stop animals from eating them. When an animal eats it, it acts as a poison and kills them because they run out of energy. The fluoroacetate occupies sites and enzymes that acetyl-CoA would occupy, and it ends up slowing down the cycle, and eventually it kills the plant or the organism. We know kind of where it exists mostly because people notice that animals eat a certain type of grass and die. Farmers complain about their livestock dying, and now it is actually used as a pesticide in certain parts of the world. Another notable example of a naturally occurring fluorinated compound is nucleosidin. Rather than a plant, this is produced by a bacteria called Streptomyces cattleya. The compound was being studied for use as an antibiotic, but it was too toxic, so it ended up not being used. But the research led to the discovery of the fluorinase enzyme. This enzyme is responsible for the carbon-fluorine bond, often referred to as the strongest bond in organic chemistry. Before that, researchers didn't know how organisms were incorporating fluorine into their systems, and this molecule led to the discovery of that. Additionally, there are fluorinated fatty acids that have been discovered, and also fluorothreonine. Structure of nucleosidin is up at the top right, and then 4-fluorothreonine is a amino acid derivative. It was discovered later. So one thing that a lot of these molecules have in common is that they're very toxic. This is a lot of the reason that organisms have a hard time using them in their bodies, and also there's not a very strict class of molecule that the fluorinated compounds belong to. Fatty acids, non-polar, fluoroacetates, very polar. There's not a certain rule that the compounds have to follow. It's found in all sorts of things. So here we have the research process. It starts with gathering plant material and identifying the species. After that, we dry it. Then we extract what we need out of it by a process called maceration. We evaporate off the solvent that's left over from the process, and then the plan was to use NMR to search for evidence of fluorine in the sample. We'll touch more on that later. So first we had to gather the plant material. There are a lot of species in Florida, over thousands. We always hear about the biodiversity in Florida, and this speaks a little to that. All the species that we use were collected from the UCF Arboretum. This is because people are more likely to know what they are since most of the specimens were brought in from other places. When I would gather the plants, I would rip them up by their roots and make sure I get parts from every part of the plant. In the molecules that have been discovered so far, they've been found in different parts of the plants, be it the roots, the leaves, the stems, or the flowers. They're not always concentrated to one area of the plant. After that, I would have to identify the plants. There's a lot of resources online for identifying them. Most of them were pretty unique flowering plants. That's why I picked them, so they would be easier to identify, because a lot of times it's very hard to identify species and subspecies of different like ferns. There are a lot of databases online that have pictures and different identifying characteristics listed for each species. There are things called dichotomous keys that you use to narrow down which species you have. So after you gather the plant material, we dried it, just placing it near a dehumidifier and hanging it up overnight. This allowed it to be sufficiently dry so there's not too much water in the extract that we get out of the plant. Then you have to isolate the extract. There are a lot of methods to do this. There's supercritical CO2 extraction, which we all did in our organic chemistry lab. There's Soxhlet extraction, which uses some glassware and a filter. And then what we did was a process called maceration, which just involves grinding up the plant and then soaking it in some solvent. A big part of this was picking the solvents we wanted to use to try to leach out what we needed from the plants. So to cover the spectrum of polarity, we used hexane, which is on the very nonpolar side, ethanol, which is kind of moderately polar, and then water, which is very polar, to make sure we got all the constituents out of the plant that we wanted. As we saw before, fluorine is found in compounds that have all sorts of polarities, so you can't really narrow it down. Another factor in deciding the solvents was ease of access. These solvents are found in any lab pretty much, and then also environmentally friendly ones we try to use. Water and ethanol you can just discard pretty easily, and hexane is easily disposed of. So the extraction method we used was maceration. We chose this over the other ones because it was simple and cheap. Yeah, you don't need a lot of fancy equipment or exotic solvents. There's also not that many steps. All you have to do is grind up the dried plant material. We used a mortar and pestle. You place it in a closed container with some solvent. We separated it out by the different polarities, and then you just let it sit for like 3 or more days. There's no limit on how long, but you need at least 3 days, and you just shake it occasionally, agitate it. The solvents over time leach the natural compounds out of the plants and leave behind essentially cellulose. After this, you strain off the liquid phase and then discard the empty plant material, and then you evaporate off the solvent. We used rotary evaporation for that. So after evaporating off the excess solvents that we didn't need, we were left with some very viscous plant extract. To prepare an NMR sample, we simply dissolved the extract in an NMR tube with deuterated chloroform. We had previously found the limit of detection of the instrument by doing a series of dilutions and calculating the concentrations, eventually until we could only detect the signal about half the time, distinguishing the signal from the noise. Unfortunately, the NMR instrument has been shut down, along with the entire campus, due to the COVID-19 outbreak, so there's no way to get any results by analyzing the samples, unfortunately. So here are some of the species we analyzed. We may have seen them around before. The one on the left, Phlebodium aureum, is just your typical fern. See it on the ground a lot around the Central Florida area. It is native. All of the species are native. We have Polygala lutea on the right. It's just a flowering plant. And here we have 2 more of the species analyzed for a total of 4. Another fern on the left, Woodwardia virginica, a commonly found fern around the area. And then on the right, we have Linaria canadensis. I wanted to include a picture from the databases they have online from the herbariums. This is what they look like. They dry the plants, they press them, and then you can use that to identify the plant species you have. They're used for research a lot, the samples in the herbariums. So now we have what we would do if the results were positive. While it is very unlikely that any of the samples contain fluorine, it's also possible that all 4 of them contain fluorine. There's no way for us to know until they are analyzed. If we did find fluorine in them, we would have to then separate the individual molecule from the rest of the solution that we don't want, just to further characterize it. We would have to use chromatographic techniques such as liquid chromatography, gas chromatography, a series of diffractions, fractionating out the molecule that we want. We would already have it narrowed down a significant amount due to which extract it was found in, be it the moderately polar, the polar, or the nonpolar. Between that and the signals that we would get from the fluorine NMR, we would have some idea of the nature of the molecule, but this would help us figure it out even further. In case fluorine was found, we have samples of the individual plants saved. It is likely that if we were trying to elucidate one singular molecule, that we would have to go out and get more samples of the same species so that we would have enough extract to find the one molecule responsible for the fluorine. Now that my time in the lab is done, there are a few things to do moving forward and scaling up the project. If it were to move forward, we would have to get better equipment. No fancy equipment was used, but in order to speed up the process, you could do so exponentially by just having equipment suited for the job. So on the bottom left, we have a multi-sock slit extractor. This would allow for faster extraction than maceration, rather than it having to sit in jars of solvent for like a week at a time. It would just take much less time and a lot less solvent by using a sock slit extractor. It would also allow for a lot of samples to be run at once. This on the right, which is just an industrial grinder, it was very difficult to grind up the samples with just the mortar and pestle. I would like to thank Dr. Elsheimer, of course, for letting me to use his lab and also helping me along the way. Dr. Elizabeth Harris helped me with identifying some of the samples, teaching me a little bit of how to use the dichotomous keys. She's also the curator of the UCF herbarium. And then also Colin Ferguson, who helped me work on the project. Here is my list of references. Thank you for watching.